What is life? It sounds simple, but in science, it's one of the hardest questions we have. Because if we want to search for life beyond Earth, as we are doing now in NASA, we first need to know what are we actually looking for. Here lies the problem. You might think life is easy to recognize, as in animals, plants, and bacteria. But when scientists try to define life precisely, the boundary begins to blur. Take viruses, for example. Viruses contain genetic material and they evolve through natural selection, as it described by Charles Darwin. But they cannot reproduce independently. They have no metabolism, and outside a host cell, they are metabolically inactive. So, are they alive? There's no single agreed answer. Many scientists avoid placing viruses strictly either category, living or non-living. Instead, they are often described as biological entities that exist at the edge of life. Some researchers even argue that viruses should be considered a part of a broader concept known as the virusphere, rather than a part of traditional tree of life. And this reveals something important. Our categories right now may be too simple for our natural world. To deal with this, scientists use working definitions. One widely cited definition comes from NASA. It says that life itself is a self-sustaining chemical system capable of Darwinian evolution. This highlights two key ideas: life being a self-sustaining chemistry and having the ability to evolve. But even this is not universal. It's a practical definition useful for research but not complete. Another way to understand life is through physics. In physics, living systems exist far from equilibrium. According to the second law of thermodynamics, total entropy in a system tends to increase over time. Life does not violate this law. Instead, it maintains internal order by consuming energy and exporting entropy to its surrounding. This idea was first explored by Erwin Schrödinger, who described life as a maintaining order through energy flow. So it means, in this sense, that life is not a static object, but a dynamic process. And this leads to a deeper question. When does chemistry becomes biology? In Miller-Urey experiment, scientists showed that simple early Earth conditions can produce amino acids, which are key components of proteins. This demonstrated that the building blocks of life can arise from non-living chemistry. Another major idea is the RNA world hypothesis, which proposes that early life may have relied on RNA before DNA and proteins became dominant. But this remains still as a hypothesis, not a complete explanation. And what is the challenge? Everything we know about life comes from a single example, the place we live in, Earth. But environments elsewhere can be very different. On Saturn's moon, Titan, for example, lakes are made of ethane and methane instead of water. Some scientists have explored whether alternative chemistries could support life in such conditions, but this still remains speculative. If we only search for life that resembles our Earth's biology, we risk overlooking unfamiliar forms. And missions right now, like the Perseverance rover, are not designed to detect life directly. Instead, what are they doing? They're searching for biosignatures of chemical or structural patterns that may indicate past biological activity. Because even after life is gone, its effects on the environment can persist. So maybe the real question isn't what is life, but what kinds of system can become life.